Hello guys, welcome back to my channel. In this video series, we are talking about the applications of biotechnology in the field of agriculture. In the last video, we touched upon how to give insect pest resistance to plant using the CRY gene, the Bt toxin gene. As I told you in the last video, today we'll start with a new mechanism of doing the same thing via RNA interference. Before understanding that, let us understand about the pest that is attacking the plant. Okay, so the pest is a nematode. Okay, it's a nematode called Melodigyne incognitia. Okay, this is a nematode. Please note, it is not a bacteria, it is not a virus, it is not a fungi. Don't write anything such in your exam. It's a nematode. Okay, by the name Melodigyne incognitia. Okay, which plant is it affecting? It affects the plant of tobacco. Okay, it affects the tobacco plant. Which part of the tobacco plant? It affects the roots of the tobacco plant. Okay, what does it cause? Well, it causes a disease known as the root knot disease. The root knot disease. What is a knot? To tell you in a simple words, a knot, एक गांठ होती है ना, वो होती है knot. Okay, so it causes such kind of galls in the root of the plant. Okay, so let's say this is my plant. Okay, this is my tobacco plant. In the roots, what is it going to form? Knots, these big galls. Okay, now what is going to be the disadvantage of this? These galls will drain out. So what are they going to drain out? They are going to drain out the products of photosynthesis. Okay, they are called the photosynthates. Okay, it's going to drain them out. The sugars and all that are produced, they will be drained out of the plant. It's also going to drain out the nutrients from the plant. Okay, in the younger plants, in the baby plants, this is fatal, but in the bigger plants, it leads to a significant decrease in the productivity of the yield of the plant. Okay, overall globally it leads to about 5% of the total crop losses. Okay, that's quite a lot. So we need to come up with a mechanism to curb this. Okay, so we'll talk how to curb this through RNA interference. Let's try to understand what is RNA interference. Okay, by the name itself, it is clear that an RNA will be involved, and interference means what? To interfere with something, okay? To prevent something from acting in its normal way. So what are we interfering with? We are interfering with the expression of an mRNA, okay? The translation will not happen. The mRNA will get degraded. We'll look at that, but let's begin from where the story began, okay? It was discovered by two scientists who were working in their lab. The names were Andrew Fire and Craig Mello. Okay, they were working with an organism called C. elegans. Okay, and they noted this phenomena in this organism called C. elegans. Okay, RNA interference is also known as co-suppression and post-transcriptional gene silencing. Okay, why? Because there is transcription of DNA to mRNA, but the translation step is blocked. Okay, so protein will not form. Why are we trying to do this? Okay, in all eukaryotic organism, RNA interference exists as a mechanism of cellular defense. It is a mechanism of cellular defense. Okay, see guys, double-stranded RNA is not normal to our systems. Okay, double-stranded RNA is not usually present in the eukaryotic systems. Let's say the mRNA or the rRNA, they are all single-stranded molecules, yes? Double-stranded RNA means that there is some kind of foreign RNA coming into the system. So eukaryotes now put up a defense against it. Where could this double-stranded RNA be coming from? See, there could be multiple sources. Let's say there's a virus that has a double-stranded RNA as its genome. Okay, so when this virus infects a cell, this DNA, this double-stranded RNA might come into the host. Okay, now second could be mobile genetic elements known as transposons. Okay, they replicate via an RNA intermediate. So from here also the double-stranded RNA will come. So as soon as the double-stranded RNA comes, our system tries to cut down the mRNA. Why? Because if the mRNA is blocked, the protein will not form and the virus infection will not spread. Let's say this is a cell. Okay. Now a virus genome is detected in the cell. Okay. So what my system is going to do, it's going to try to block the mRNA and protein formation in this cell. So there is no protein formation in the cell. This cell will not grow. Okay. So the virus infection will be restricted to this cell only. Okay. Proteins are what? They're enzymes. They are very critical for the normal functioning of any organism. Okay. So if I block the protein, this cell is going to die eventually and the virus is going to die along with it. Okay. The infection will not spread. That is why it is a cellular defense. Now in the next step, we are going to look at what is the mechanism of RNA interference. Begin looking at the mechanism of RNA interference. Okay, I'm not going to tell you all the steps, but briefly I'm going to run you through the mechanism. Okay, so our story begins with, like I told you, double-stranded RNA. What is double-stranded RNA, guys? Double-stranded RNA is that RNA that has two strands, right? Two strands. Okay, two strands of RNA. Okay, this is my double-stranded RNA that was detected in my cell, in my system. It is a foreign RNA that my eukaryotic system is putting up a defense against. Okay, so this is this molecule. Now what is going to happen is an enzyme called the dicer enzyme. Okay, I dice vegetables, I chop vegetables. Okay, dice मतलब काटना. Okay, so एक ऐसी एंजाइम है जो कि इस मॉलिक्यूल को क्या कर देगी, काट देगी. It's going to break it down into smaller pieces. Okay, so we're breaking both the strands down into smaller pieces. Okay, fine. This is cleaved into smaller pieces. What are these smaller pieces called? They are called siRNA. Si stands for small interfering RNA. Okay, they are small pieces of RNA that are normally 20 to 25 nucleotides long. Okay, so my dicer will form siRNA. Okay, so I'm going to cleave this. In the next step, what is going to happen is a complex is going to form. RISC. The complex is called RISC. RISC stands for RNA induced silencing complex. Okay, RNA induced silencing complex. Why? Because the double-stranded RNA is inducing the silencing of my mRNA. Okay, so it is called the RISC complex. Okay, so my RISC complex is going to come. What is it going to do? Okay, in my siRNA, guys, there are two strands. In my siRNA, there are two strands. Okay, fine. Both these strands have specific names. Okay, the first strand is called the passenger strand and the second strand is called the guide strand. Okay, guide strand. My RISC complex is going to remove the passenger strand from the small interfering RNA. We'll only be left with the guide strand. Now towards the end of this mechanism, I'll tell you why they're called so. Okay, but for now, let's move ahead with these names. Okay, so now my RISC is going to cleave away the passenger strand and I will be left with what? The guide strand. Okay, so RISC removes my passenger strand. Okay, now what is this? This is a single stranded RNA. Because why? We are seeing only one strand of RNA here. Okay, the passenger strand has been removed. Now in the next step, another protein called the Argonaute protein is going to come. And it's going to simply bind with my single-stranded guide strand RNA. Okay. Now this Argonaute protein is going to do what? This complex is going to specifically go and bind with my mRNA. Now guys, this is going to come and bind Okay, let's draw the Argonaute partner as well. Okay, now what is going to happen? It has bound specifically, see here, with the mRNA. Now it is going to cleave off this mRNA. Okay, the mRNA is cleaved. Cleaved means it is chopped down into smaller pieces. Further, it will be digested by the enzymes of the cell, and this mRNA is going to be removed off from the cell. Okay, this is the mechanism. Now guys, like I told you, now let's talk about what is passenger and guide strand. Okay, see, guide का क्या मतलब होता है? किसी को दिशा दिखाना, किसी को बताना कि कहाँ जाना है. Okay, that is guiding. To give directions is guiding. Okay, now see, this strand, the guide strand, is complementary to my mRNA. So it is guiding it towards the mRNA. Okay, it is telling where to bind. That is why it is called the guide strand. The guide strand is complementary to my mRNA. Okay, the passenger strand has the exact same sequence as my mRNA. It's not going to bind and it's going to be removed. So this was the mechanism of RNA interference and how this mRNA will be cleaved into smaller pieces. Now let's move on and look at how it is going to save my plant from the infection of the nematode.